In this video, you will learn about the sensory transduction mechanisms and pathways for two of our special senses that involve chemoreceptors. These are olfaction, our sense of smell, and gustation, the sense of taste. Let's start with olfaction. Olfaction is our ability to detect airborne chemicals, what we call odorants. When you inhale, odorant molecules dissolve into the mucus of the olfactory epithelium inside your nasal cavity. These molecules bind to special G protein-coupled receptors located on the dendrites of olfactory neurons. There are about 350 distinct types of olfactory receptor genes, and each one codes for a receptor that recognizes different molecular shapes. Even though there are only 350 receptor types, we can distinguish many more different odors because each odorant can activate one receptor type or combinations of them, giving us a huge variety of smell perceptions. When an odorant binds to its receptor, the G protein inside the neuron is activated. That G protein detaches and activates an enzyme called adenylyl cyclase in the cytoplasm. Adenylyl cyclase then produces a second messenger molecule called cyclic AMP. This cyclic AMP opens ion channels, allowing sodium and calcium ions to enter the neuron. The influx of these positive ions depolarizes the olfactory receptor cell's membrane, and if the depolarization reaches threshold, this neuron fires an action potential. These action potentials travel along the olfactory nerve, or cranial nerve number 1, into the olfactory bulb, where the olfactory neurons synapse with mitral cells. From there, the axons of the mitral cells form the olfactory tract, a white matter pathway that carries information posteriorly toward the brain. These signals arrive at the primary olfactory cortex, located in the temporal lobe. This is where we become consciously aware of the smell and can identify it. Interestingly, olfaction is the only sensory pathway that does not synapse in the thalamus before reaching the cerebral cortex. From the olfactory cortex, signals are sent to the amygdala, hippocampus, hypothalamus, and other limbic system structures, areas involved in emotion and memory. That's why smells can trigger such strong emotional memories or even visceral responses like salivation, sexual arousal, or nausea. Now let's move on to gustation, our sense of taste. Taste begins when molecules dissolved in saliva activate specialized receptor cells located within taste buds. Raised bumps on the tongue called papillae contain the taste buds. There are 4 types of papillae based on their appearance: circumvallate, foliate, filiform, and fungiform. Within the structure of the papillae are taste buds, clusters of cells that include the specialized gustatory receptor cells. These receptor cells are sensitive to the chemicals contained within foods that are ingested, and they release neurotransmitters based on the amount of the chemical in the food. Physiologists have identified 5 distinct tastes: sweet, bitter, savory, salty, and sour. Each taste type has its own unique transduction mechanism, but all eventually cause influx of calcium ions into the gustatory receptor cells. This calcium rise triggers neurotransmitter release onto the dendrites of afferent sensory neurons. Let's go through each taste type. For the sweet taste, sugars like sucrose, lactose, fructose, or glucose bind to G protein-coupled receptors on the receptor cell membrane. Bitter compounds, such as alkaloids found in coffee or chocolate, also bind to G protein-coupled receptors. For the savory taste, amino acids, especially glutamate, bind to similar G protein-coupled receptors, creating that meaty or broth-like taste. For salty and sour, the mechanism is a little different. It's ion-based rather than receptor-based. The salty taste results from sodium ions entering directly through ion channels, depolarizing the gustatory receptor cell. Sour taste results from hydrogen ions entering through ion channels to depolarize the gustatory receptor cell membrane. Once these gustatory receptor cells release neurotransmitters, the afferent neurons carry the information toward the brain. These neurons travel through 3 cranial nerves: the cranial nerve number 7, the facial nerve, number 9, glossopharyngeal nerve, and number 10, the vagus nerve. These afferent fibers synapse in the solitary nucleus of the medulla oblongata. From there, some neurons activate parasympathetic reflexes, such as stimulating saliva production. Other neurons relay the signal to the thalamus, and from the thalamus to the gustatory cortex, located in the insula and an adjacent portion of the parietal lobe, where the brain perceives and identifies the sense of taste. To summarize, olfaction uses G protein-coupled receptors to detect odors. The olfactory nerves carry the signal to the olfactory bulbs. Then, olfactory tracts send information directly to the temporal lobes. Gustation uses both G protein-coupled and ion channel mechanisms to detect the 5 basic tastes, with information traveling through cranial nerves 7, 9, and 10. This information is first relayed to the thalamus before reaching the gustatory cortex in the insula and parietal lobes. Together, these senses help us enjoy food, detect danger, and even trigger powerful emotional memories.